Hello, everybody, and welcome to the Ultimate Health Model podcast, where we take a deep dive into uncharted waters on everything health. Today, I have the distinct pleasure of welcoming Michael Snyder, who is a genomics researcher, professor, head of the Snyder lab, right, at Stanford University. I wasn't sure. Were you part of the genomics project for the mapping the genome?
Well, we didn't actually determine the sequence at our lab, but we've been doing a lot of work to try and understand the function, meaning where are all the genes, how do they work, when do they express, and
Okay.
what issues, that sort of thing. So the next step, understanding how the genome works, our lab was a big part of that.
Nice. So, Michael, why don't you, for our friends listening, why don't you kind of go through your a little bit about your background, how you got into this. I know I find this conversation is going to be really fascinating because genes are basically the blueprint of life, and the fact that for our friends listening, there's something called the DNA molecule. Probably you guys probably understand what that is. I'm not sure, but that's the DNA molecule. It has these pairs, these little things on them like rungs, and there is 3 billion of these rungs, or I guess one half on each side. Anyway, I thought to start with that. There's 3 billion base pairs. That's You can't even comprehend that kind of number, right? Michael's laughing because it's true. It's like when you really think about how complicated, incredibly complex, complicated, incredible the body is, it just blows my mind. So why don't you tell us a little about your story, Michael, how you got into this, and all that kind of stuff.
So anyway, I guess my background is I've been around for a little while. We've got involved in when it first became possible to isolate genes and study them way back in the 70s, believe it or not, late 70s. Became possible, and that led to ultimately being able to sequence people's DNA. And so we're involved in some of the early trying to characterize genes. The genes now, let's back up. So as you point out, our DNA is our blueprint of life. It's a dictionary. It lays out all the, if you will, letters which turn into words, which turn into sentences that basically make your basic program. And that had been uncharted when we started this, and some methods came along for being able to try to actually start studying small snippets of that. And then later projects have evolved to be able to characterize the whole thing, your whole set of DNA, believe it or not. And it is a big challenge, as you point out. We have 2 copies of 3 billion of these
Wow.
letters. It's just
Wow.
incredible. And so it was a big challenge. It needed new technologies, new methods. And so our lab, in fact, did invent some of these. And then the group went on to actually sequence the first, if you will, reference genome, just a sort of representative genome, if you will, which is essentially your collection of DNA. And that really laid the groundwork for this whole area. But then a big challenge came. So you have, as I say, 3 billion these letters, all structured a particular sequence. And believe it or not, we're all very, very similar to one another.
Yeah.
Only by 0.1%.
Yeah, I read that in your book. 0.1%.
Yeah,
It's only a million. It's only a million. That's what it's only a million of the bases or something, right? Out of 3 billion.
Right. That's correct. Yeah, 3 million. So it's very, very small number. So the difference between you and me is not all that much
Yeah.
at a genetic level. Now, those 3 billion bases, they encode about, depends how you want to count it, 20 or 40,000 genes, which in turn, 20,000 genes encode proteins. And those are the things that do your work. They digest your food. They make the things on your skin that keep you safe. And, your eye has proteins that help you sense light and all these sorts of things. So genes encode proteins that do the final business. And there are 20,000 of those, although you can get lots of variations of these proteins. And so we did a lot of work then to map out where are all these genes. First of all, how many are there? People used to think there were 100,000. Well, it turns out there was a lot less, although the gene cells are a little bit complicated. And then in turn, they make, as I say, essentially 20,000 proteins. And then we've been characterizing how the genes are turned on and off as well. That's a big deal, meaning your skin proteins are only made in your skin, and your digestive proteins in your stomach and intestine. They're expressed in very specific places, and there's a complicated machinery that makes sure that happens and all works right. So anyway, we got involved in doing our lab was pretty fundamental. We invented a lot of the methods for being able to tell where the genes are and where all those control sequences are for turning genes on and off. And so we've been involved in this genome characterization. And then more recently, we've been involved in trying to understand how to apply this to human health, like what kinds of genes get mutated to lead to what kinds of diseases. And a classic example that many listeners may be aware of is that women who have breast cancer running in their family often have mutation in a gene called BRCA. And there's a famous case of Angelina Jolie. It turns out her mom died at age 51 of breast cancer, and she wanted to know if she had it. Sure enough, she actually has a mutation in this gene, and she, in fact, had her breast removed as a way of preventing getting breast cancer. So
Let me stop you for 1 second right there, so I can clarify things. So when you're the BRCA gene, so our friends listening, what that means is that on these base pairs, there's 4 letters that they use, basically C, G, A, and T, and they're different nucleotides, guanine, and the other one, I don't remember their name, but there's 4 nucleotides. And so I believe what you're saying then, Michael, is that if it's supposed to be C, G, A, T, 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 let's say it could become C, G, A, T, C, or so it's a letter goes out of place. Is that correct?
Yeah, that's correct. Or it could even get deleted or an extra one added. That screws up the whole gene. Okay. And that causes a problem, often kills the gene, so to speak.
Okay, so it kills the gene. And then how do you know what's really fascinating? So, and besides what they name it, how do they know on the long billion, 3 billion long, nucleotide strand, how do they know where that gene is about breast cancer? I don't understand. That's really very hard for me to understand.
Oh, that's a great question. And so what they do is they they go into a lot of families that have this problem where the mom had breast cancer. And so imagine 1,000 families, and they go in and then they sequence their genes, if you will, and they can
match them up.
That's the one thing they have in common. Gosh, you look at 1,000 families, next thing you know, basically 100 of them will have, well, it's not quite 100. Let's say it's 50 families will have a mutation in this gene. That's way more than by chance. Okay.
I see.
So it's one, they call them association studies. You can show that when this gene is mutated, it's associated with this gene, with this disease. And so that's been done for breast cancer. It's done. Have you heard of cystic fibrosis? Where problems in their lungs, same thing. They have a problem in a particular gene. It's called the CTCF or the cystic fibrosis gene. That's probably the easiest way to call it. And so it's mutated, and those individuals that have problems in their lung, they have a problem in an ion channel that
see. Let me ask you a really interesting question then. So, if that's the case, there's DNA repair systems going on all the time in the cells and in the DNA molecule. Why can't, and for our friends listening, this might pique your interest, why can't they just take the DNA repair system and add or detract or subtract that particular letter to make it right?
Yeah. Well, so naturally, that happens, as our cells divide and what have you, they most times they do get fixed. Now, as far as us going in and fixing problems, that's been hard. And in some areas, it's now possible. It's just emerging now. We can now go in and make those changes to try and fix things. It's hard to do that. Like, imagine you have, believe it or not, 37 trillion cells.
Yeah, I know that. Yeah.
That gene is essential for every one of your cells. It's hard to do that for all your cells. So what they're doing in some cases, like you've probably heard of sickle cell anemia, in those cases, that's a blood disease. There you can take out the cells that are in your bone marrow that make your blood cells. And so
Yeah.
you can actually now go in and make the change like you're describing and fix people. That's only just become possible. First, we had to show the methods were safe. You don't want to try and fix things and then make mutations elsewhere. And that has happened where they try
Oh.
helping
And
people out. Go ahead.
how exactly do they do that? So if what you're saying is they can go in there and they can take that letter and put it the right way. So like, let's say it's for our friends listening, let's say, because the DNA is like an alphabet, it's like a language. And like we talked about earlier, their sentences, their letters, their paragraphs. And so when you read it, so it would be like, the word you or your, Y-O-U-R, had said Y-O-U-S or something. So the word doesn't make sense. So we
Right.
go in and we put the R back in there and then the word makes sense.
Yep.
So for my question is, why can't they just you're saying they're doing it now. So can't we can they make all humans impervious to disease? Because you go the BRCA gene for breast cancer, the sickle cell anemia one, the cystic fibrosis. Can we go in there and just do all of them and we're all fine?
Well, in certain cases, they're now just able to. First, you had to make sure the technology works because it's not trivial to do this. And they now have pretty good technology. It does work, meaning what you don't want to do is say, I fix that S to an R, and suddenly I made a mutation somewhere else unknown that created a T and actually was worse for the person, caused cancer. So get it all working properly. And that's taken, believe it or not, at least a decade, if not longer. First, it was getting technology in the first place. Then there's improving it to the point it works. So that works and works mostly just changes that change you want to make. That's step one. Step two is you can only really change it in systems that are accessible. So I mentioned sickle cell anemia. You can do that because I can fix the bone marrow and that'll fix the blood cells. You see, that'll actually fix the disease. But imagine it's a brain mutation.
Uh-huh.
How are you going to get in there and change all your brain cells? That's not so easy, right? You can't just take them out, grow them, whatever. People are working on it, but it's a lot more challenging. Does that make sense? It's just that some area.
Yeah, that makes sense. But I'm confused because if DNA might help me understand this, Michael. If DNA is the blueprint, they take just DNA from anywhere. They find that gene, it won't translate to all the brain cells once it's put back, is what you're saying.
Yeah, you'd have to get in there on every single cell and try and change it. You see,
Why is that though? Why is that if it's the DNA is the same in every cell and you change that one mutation?
Because I'd have to go in. Imagine I'm trying to change this in your brain. I'm
Yeah.
going to change billions of cells specifically. And these cells aren't that easy to get at. They're not dividing. Okay. There's no good technology for me to get in there and change every one of your brain cells.
I see. Because I thought what you're saying is that if we take that mutation, make it right, it automatically changes all the other cells. That's not correct.
No, unfortunately,
Okay.
unfortunately, no. But for the bone marrow, see, like for the sickle cell, I can pull out the bone marrow, I can make the change, and then I can grow up the cells that have the correct copy. Do you see?
And put them, okay, yeah.
And I put that back. So, and that will swap out all the other cells and become fine. So it's because I can make the change and then isolate that and grow it up and make a pure population of fixed cells.
I see.
I can fix the person. But if the cells aren't dividing, it's very hard. There's a lot of challenges still. People are working on this and I'm optimistic in the future, but we're decades away from changing brain diseases with genetic engineering.
I see. So we're decades away from being superhumans.
Yeah. Now there are interestingly enough, imagine I know that you're at risk for something and you're going to have a kid. Well, I can go in with in vitro fertilization,
okay,
and then go in and change the embryo or early on. Normally you would just select for an embryo that doesn't have that and then implant that back into the mom and get a good kid that way. But in principle, people will work on trying to change these genetic diseases that affect all your cells. You're going to have to do that very early. And we're not ready for that yet. We're getting there, but we're not
that makes sense. So,
Someone who did this, by the way, and he got in trouble. He,
oh,
he was trying to make mutations that would help. Um, Basically, it was the AIDS receptor, and he was making change so these kids wouldn't get AIDS, basically. And that was very controversial at the time. Most people thought it was a bad idea because it wasn't clear the technology was safe yet, and that is a big deal. Making
that makes sense. That makes sense. So, for our friends listening, I want you to explain, if you could, Michael, how does so when I was reading your book, and it's funny because just, full disclosure here, when I have podcasts, I've got to do a lot of research to learn about my guest and what they know. And I knew nothing about genetics. I knew about DNA. Yeah, I knew about DNA and this. I go, oh God, I got to be smart enough to hold my own with Michael. I got to learn some things here. And reading your book, man, a lot of them over my head. But I did learn some things about,
It's hard to hear that, Ben, because I was trying to write it for the layperson. So,
Michael,
I got a ways to go.
yeah, no, that did not. Unfortunately, that did not work. I'm sorry. I was reading I read like one page like 5 times. Like, what is he trying to say here? I couldn't. So I got the gist of all of it, but so it's the 5 billion base pairs for friends listening. That's kind of the letters, yeah. And there's something called non-coding DNA and variants and spaces. Basically, if there's only 20,000 genes, then a lot of the DNA doesn't code for anything, quote unquote. And now in the last few decades, they understand that that's not true. What is the non-coding? And for our friends listening, can you explain, does that mean and I don't even understand this myself, does that mean the letters go A, G, C, T, C, and then there's no letters, or there's O's or something, and then there's more letters? No, because I don't understand it. It's very complicated.
No. So you have letters for all 6 billion bases, but only about 2% of those bases encode the genes that make proteins, as I was mentioning before. And it's just they have a very specific sequence that leads to very specific proteins that get made. But upstream, those letters still work. They're just not coding for proteins. They're the control sequences for turning your genes on and off. And it turns out we have more of our DNA is devoted to controlling how those genes are turned on and off versus actually what the genes themselves. Does that make sense?
Yeah, it makes sense. So it's almost like, for our friends listening, it's almost like, and I wrote this in my book, that proteins are machines.
They are.
So, yeah. So if it's a machine,
molecular machines, that's
exactly what they are. I write that in my book. If it's a molecular machine, you can't just have a machine sitting there without instructions on how to use it.
You got it.
So that's what for our friends listening, when people talk about junk DNA and non-coding and they don't think it does anything, that's how those are the instructions on how to use it.
That's right.
So for our friends listening, we you guys as scientists and geneticists, they're working on. They don't even understand all the non-coding stuff yet, right? Is that correct? On how these things work?
We know way more than we used to, but we still have a ways to go. Yeah. So absolutely. So in the end, it's kind of surprising, and I think that's one of the things that came out of our work, that we actually devote more of our DNA to turning the genes on and off. Genes themselves, which is a surprise, I think, most people. That's just how it is. I wouldn't mind talking a little bit about how DNA gets modified or gets.
Yeah, absolutely. Yeah. Whatever you want to talk, because it's all fascinating to me.
Yeah, no, I think.
And try not to go over my head if you can, or our listeners, because I'm very smart, but still, I'm like, okay, I got to go through that 5 or 6 times. So I don't understand it.
Well, that's why I'm on the show, though. I can. All right. Anyway, what I'd like to impress upon your listeners is that, so your DNA is the blueprint. It gives you the basic blueprint. Like identical twins have nearly identical DNA. But how the DNA operates is these genes getting turned on and off. It turns out your lifestyle can modify your DNA.
Yep.
So even though you're going to have two people who are identically the same, they actually can turn out a little bit different. And the analogy people like to use is music. You can have score for music, but how you play that music can give a very different product. And I'll date myself a little bit, but Louie Louie, that was.
Of course I know Louie Louie.
So that song, there's like over 50 renditions of that. And they're very different. They all have the same lyrics, right? And the same general music, but how it gets played is very different from one group to the next. That's one way of saying this. And that's the power of epigenetics, meaning modified DNA. And it turns out that the food you eat, exercise can impact that.
Yeah, and I just quickly, I don't want to interrupt you, but for friends listening, and that's exactly what we're both in agreement on. I wrote a book too about health, and one of the big factors, and you're shaking your head, that's so important that you can't. So genetics play a part, but if you can't go eating a bunch of cookies, cakes, be stressed out, don't sleep, have a horrible, stress and all these things, and expect that, oh, well, all my genes there's no mutations and this and that, and I'm not gonna get this because my gene. No, it doesn't work that way. They work together. And that's what people, our friends listening, need to understand. Do not, and I can underscore this enough, do not underestimate the environment in controlling your genes and your destiny, so to speak.
100% agree. And maybe to add to that, so your DNA accounts for, you can show this genetically, 16% of your lifespan. Okay, meaning the rest comes from non-DNA, these other things. So that means your lifestyle and environment accounts for more of your lifespan than your DNA per se. Now, your DNA is still very, very important. Don't get that wrong, but it's just one factor in the big equation.
And also for our friends listening, understand that when you have a bad lifestyle, let's say you're smoking, you're stressed out, you're eating bad food, what that's going to do is that's going to cause inflammation, and that inflammation causes reactive oxygen species. So what does that mean? That means they're molecules that are very reactive and damage things like DNA and lipids in the cells and proteins, and all that can do damage to the DNA. And then you have what's called repair enzymes, DNA repair enzymes, proteins that go and repair that. But like Michael's saying, if you constantly damage that DNA from your bad lifestyle, the repair mechanisms are going to wear out.
Yeah, no, it's I 100% agree. And I can even take that again one step further. Smoking has agents in it that will directly modify your DNA, not even through reactive oxygen. I mean, they can. There are components in smoking that make reactive oxygen species, but there are something called alkylating agents in smoke that will go right in and directly modify your DNA. And that's why it's so terrible.
Wow. So it actually directly changes the expression of the DNA.
But you're right too. Watch out for inflammation, anything causing oxidative stress, which inflammation doesn't do, but other things can cause oxidative stress as well, can cause these problems, just like you're saying. And so it's very, very important to keep that under control.
Yeah. And like I said, it's just to reiterate this, it's so important. Please don't think that your genes are everything, because you have to have a good lifestyle, and which includes, like we talk about, and it's other things too that people are not aware of, Michael, which I think is very important. And let's quickly go over these for our friends listening. We all talk about diet, how important diet. We all know that, unless you're living under a rock. We all know about stress. We all know about sleep. But there's stuff like grounding and connecting to nature, which is, cutting edge, kind of research and science, and sunlight, the right kind of sunlight, from the, morning sunlight and the dusk sunlight, a lot of these things also help the DNA, the way you breathe.
I even know how it works. Like sunlight helps you make vitamin D.
Yeah, there you go. And which helps too with DNA. So all these things. Go ahead.
Right. Well, I was going to say, there's a lot of things we don't understand how they work. You may know in Japan, they do nature bathing.
Yeah. It's called force bathing, right?
You got it. And which is very interesting. And it's thought there are important compounds made. We don't fully understand this. This is an area that our labs, believe it or not, working on to try and sort this out. But yeah, presumably it makes compounds that actually help your body. Maybe it's just helping reduce stress and oxidative stress and things like that. But that's an area of high interest.
Yeah, that's very interesting. And speaking of the oxidative stress and the force bathing, from the research I've done, is that the stress causes, like you said, the oxidative stress causes the inflammation, which causes mitochondrial dysfunction, which causes more inflammation, which causes immune system suppression. So all these things go around and around.
Right.
And so when you do the wrong things, it might not necessarily directly impact the DNA, but if it impacts let's say it impacts the mitochondria and damages the mitochondria. The mitochondria is responsible for cell apoptosis. So then that's going to create problems with, and that plus obviously all these things signal together. So if you damage the mitochondria, then you might be damaging the nucleus, which is damaging the DNA in the nucleus. Not to mention, for our friends listening, the mitochondria has its own DNA.
Yeah, that's right. The mitochondria is very important. It's kind of interesting too, maybe to add to what you're saying. As you get older, your mitochondria will accumulate this DNA damage.
Yeah.
And then it's thought that, at least we think, it definitely is responsible for fatigue and as you get older. So that's one thing that again, taking antioxidants should help with that and reducing oxidative stress. So I think it's one area that if we all want people to live long, healthy health spans, basically. You want your health span to equal your lifespan.
Yeah, of course.
And one area that's going to be tricky, I think, as we get older, if you want people to live forever, you're actually going to have to swap out their mitochondria. So I think, but that's very true.
Yeah, and that's interesting to say that, because I was reading an article, watching this video. This guy was talking about mitochondria farming. And God, I don't remember all the aspects of it, but for our friends listening, look up mitochondria farming, mitochondrial banks.
What companies is doing is figured out how to make lots of mitochondria.
Yeah.
They're very interested in solving this problem.
Yeah, and so they're basically making mitochondria in like these banks of mitochondria, almost like blood banks. You go get mitochondria, and theoretically, that could improve health and maybe longevity, and these mitochondrial banks. And also, I found fascinating about mitochondria that they can actually be transferred between cells. I don't know if you know about this, Michael, but I was reading. They're not called isolates. What are those things called? Rootlets or something? Well, of a neighboring cell needs mitochondria. It will send us a chemical, messenger signal to that cell, and it will literally take that mitochondria out of that cell in a exosome, whatever, and go through the bloodstream to the other cell. I read that. I was like, the cell is actually giving mitochondria to its neighbor and back and forth. I'm like, what the?
Yeah,
that's incredible.
Mitochondrial transfer is really quite powerful. I agree. By the way, I'll add one more thing too, which is, so we can now measure, maybe this is a little shameless plug, but one of my companies called IOLO, IOLLO, they actually will do a metabolic profile on you, meaning you give a little drop of blood, you take it from your shoulder, mail it in, and they'll measure 600 metabolites. And they can measure your oxidative stress, your inflammation, all these things.
Wow.
And so it's no longer just a matter of saying, exercise more, eat better, but they can actually see in more detail what's going on. And then people can actually act on that. And here's where you use AI to basically collect information that's both in the scientific literature and about you and make specific recommendations. Like if you're off in oxidative stress, eat blueberries and certain other things. And if you're doing some of these things already, because they can track this, you can sort of write, tell you what more to add and what to take away to improve your metabolic health. And we can do this now, I think, for many different, we call them wellness areas. And the obvious ones people are familiar with: inflammation, oxidative stress. But there are things around like your kidney function, heart, whatever. All these metabolites have, their signatures of your overall body health, if you will. It's something related to you. You need your whole body working together properly to be an efficient, well-functioning human being.
Yeah.
By measuring all these different areas. We're big on measurements. I'm a big believer that we should be measuring people deeply and tracking them, so when things go off, you can catch that and get it corrected. Kind of like your car.
Yeah, let me ask you a question about that. So if you're measuring oxidative stress or inflammation levels or C-reactive protein, especially oxidative stress, isn't depends on it's a snapshot in time. So it's not going to like, I know that the HbA1c, the hemoglobin, for your what do you call that? HbA.
Yeah, HbA1c for your
yeah. So measure, but that's a 3-month average measurement approximately of how glycated the red blood cells are. The oxidative stress measurement or the inflammation ones, how, if folks listening might want to understand this, how does that, is that a snapshot for 1 second, 1 minute, 1 hour, 1 day? I think that's an important question to ask.
Right. So the company I mentioned before, those are snapshots. We're saying try them every 3 months. So you can see, and to do what you're saying, to try to improve your markers. And 95% of people who actually take the test improve their markers by the time they do the second one, because again, they're very specific recommendations. Now, having said that, for glucose monitors, you may know now there's something called continuous glucose monitor. Right on, people, they take measurements every 5 minutes. So they can see exactly how you're responding to which foods. And it turns out different people respond to different foods differently, meaning you might spike to potatoes, I might spike to pasta, some people brown bread, some of the white bread. We're all different. And we don't fully understand the basis of that. Part of it's due to what's called your microbiome, the microbes in your gut. And so we know that's very, very important, but that's not the whole thing. Our lab's trying to figure this out, as are other labs. But those monitors are super, super powerful, these glucose monitors. So I'd highly recommend anybody try one of those. You'll never eat the same afterwards, because you'll see what's
what's spiking your sugar. Yeah, but back to the other question. I don't know if it was answered correct. Yeah, it says, what I'm saying is that you say you measure it one time and 3 months you measure it again. My question was originally, how often do these things change? Is it by minute, by second? So how do you know? So like 3 months later, how do you know all those other time in between, it was going up and down or left or right? How do you know? Does that make sense what I'm saying?
Yeah, very much. So all I can say is we don't know how to measure people, but we're recommend starting with that. Now, it's generally the case if you get people after they, first thing in the morning before they've eaten, called fasted. If you sample them and look at them, that's fairly consistent. Okay, so unless you're going off, that's kind of what you're looking for. You're trying to see if you are going off so that you can make
a general measurement, basically.
Yeah. So so these are deep profiles. They'll measure you. And if something looks like it might be going off, well, get yourself measured more frequently, I would argue, and make sure that you are getting your health under control. It's a lot easier and more important to keep people healthy than wait till they get disease and try and fix it. That's almost always impossible to do.
And that's just the whole. That's the crux of the conversation here is genetics are important. Your lifestyle is important. They're both important, period. But, you can't just expect to, like I said earlier in the conversation, you can't expect just to do whatever you want. And you have, your genes, you have no genes, mutated genes for diabetes, and expect not to get diabetes. Because as you write in your book, people have this mutation for something, type 2 diabetes and they never get it. Or people don't have the mutation for it and they do get it.
Yeah. Yeah, we don't
So,
have it all figured out, but we're getting there. Now, some things are pretty predictive. I mentioned BRCA earlier, so those you want to watch out
you want
for.
to watch it. Let me ask you a really interesting question. For our friends listening, they might want to know this too. How do you guys come up with the names for the genes?
Oh yeah, some of them are pretty quirky. A lot of genes were discovered in Drosophila, those folks, they were proud of the fact they would make strange names for the final Drosophila fly would look weird, and they'd name the thing after like Rebecca.
Oh,
So they might look like a vegetable, and they'll give it a vegetable name. So that's how some of the names come. Some of them are more. Cystic fibrosis means it's fibrosis in your lungs. So some of them make sense and match up, and some of them, although medicine in general, you'll notice that often when they diagnose something, all they're really doing is giving you the name of a condition. You can see they don't really know how it works.
okay.
Yeah. So, you have so-and-so disease. Well, all they've done is giving you the label that disease is called. They don't necessarily know how it works.
Oh, wow. Well, no, because like you said, the BRCA gene, and then also with when I watch TV and they have the drugs for breast cancer, and it's that if you have an HRF1 gene or if you're HR5 positive or negative, I don't even know. There's so much. It's so complicated. Are you HR2? If you're HR2 negative, don't take this drug, right? And I'm like, so that's what I'm saying. It's a lot of letters, right? They use a lot of letters just to kind of represent things.
Well, unfortunately, yeah, there's a lot of that kind of lingo, especially in immunology around your immune system. Yeah, they have a lot of these CD45 and these names.
Yeah. And I'm like,
Their
I
names,
can't keep track of them all.
even most scientists can't keep track of
Okay,
that.
good. So I'm not alone. Okay, so I try to make things simple. My book is like, that's what you do, that's what you don't do. I don't know the names of the genes, but here's a really simple blueprint. Anyway, I know you got to go here, but let's, for our friends listening, any parting words, and why don't you tell us how people can find you, more about your work, and all that kind of stuff, Michael?
Sure. Yeah. Well, if you're able to somehow post a link with my website, you're welcome to do that with the lab website. That'll get you there. We run research.
Yeah, we'll do it on the editing. Yeah.
Welcome to participate in it, and you can reach out to me. I'm M as in Michael, P as in Paul, and then Snyder, SNYDER, at stanford.edu. And so I generally answer my emails. And if you would like to learn more about any of our research or companies that we spun off our lab, see, what we're a little unusual. We do a lot of basic research, and then to get it out to the world, we spin off companies as a way, like this one I mentioned, Iolo, to do the metabolic. There's another one called January Eye that lets you do CGM testing, which is kind of this continuous glucose. So that's our way to try and get it out to the broader community and the world. And that's how we hope to improve people's health.
Well, awesome. Any other parting words?
Stay healthy. Track yourself. Don't wait to go to the doctor till you're sick. Get yourself checked all the time.
Yeah, get yourself checked. And
by the way, see my smart.
Yeah, you got it. Yeah, and I think too, for our friends listening, is just use your common sense too. Use your common sense. It's great we have technology for measuring genetics and stuff, all these things, but we've been around as a species for 300,000 years. And 100,000 years ago, they didn't have gene sequencing, and people were living. And so if you understand that nature is a really, really incredible thing, and the DNA molecule is just absolutely astounding and fascinating, and work together as far as nature and science and all these things. If we work together, everything will be a lot better.
You got it.
All right. Thank you so much for your time, Michael.
Thanks for having me here.